Breakthrough in serotonin receptor research. Next-gen mental health drugs explained. Imagine a future where mental health medications are not only more effective, but also act faster than the current options available. This vision is becoming a reality thanks to groundbreaking research from the Icahn School of Medicine at Mount Sinai, which has unveiled new insights into the workings of a vital brain receptor. This discovery could significantly influence the development of next-generation antidepressants and antipsychotic drugs. The study, recently published in the online journal Science Advances, delves into the 5-HT1A serotonin receptor, a crucial component in mood regulation and a common target for both traditional antidepressants and innovative therapies like psychedelics. The research paper is aptly titled Structural Determinants of G-Protein Subtype Selectivity at the Serotonin Receptor 5-HT1A. Despite its importance in clinical settings, the 5-HT1A receptor has remained somewhat of a mystery, with many of its molecular and pharmacological characteristics not fully explored until now. Senior author Daniel Wacker, PhD, who serves as an assistant professor of pharmacological sciences and neuroscience at the Icahn School of Medicine, likens this receptor to a control panel that regulates how brain cells respond to serotonin, a key neurotransmitter that influences mood, emotions, and cognitive functions. He explains, "Our findings illuminate how this control panel operates, what switches it activates, how it modulates signals, and where its limitations lie. This enhanced understanding could lead to the creation of improved therapies for mental health issues such as depression, anxiety, and schizophrenia." Utilizing advanced laboratory techniques, the research team discovered that the 5-HT1A receptor is inherently predisposed to favor certain cellular signaling pathways over others, independent of the specific drug targeting it. However, the strength of these pathways can still be influenced by the drugs used. For instance, the antipsychotic medication asenapine, known by its brand name Sapharis, was found to selectively activate a particular signaling route due to its relatively mild activity at the receptor. To gain deeper insights into these mechanisms, the researchers employed a combination of experiments conducted on lab-grown cells and high-resolution cryo-electron microscopy, a state-of-the-art imaging technique that allows scientists to visualize molecular structures at nearly atomic resolution. Their investigation focused on how various drugs activate the 5-HT1A receptor and how this receptor interacts with internal signaling proteins known as G-proteins. The different signaling pathways governed by the 5-HT1A receptor are associated with various aspects of mood, perception, and even pain. As researchers gain a clearer understanding of which pathways are activated and the mechanisms behind them, they can design drugs that more precisely target specific symptoms or conditions while minimizing unwanted side effects. Audrey L. Warren, PhD, the first author of the study and a former student in Dr. Wacker's lab, now a postdoctoral researcher at Columbia University, states, "Our work provides a molecular map of how different drugs push buttons on this receptor, activating or silencing specific pathways that influence brain function. By comprehending exactly how these drugs interact with the receptor, we can begin to predict which strategies might lead to more effective or targeted treatments, and which ones are less likely to succeed." This is a significant step toward crafting next-generation therapies that are more precise and have fewer side effects. In a particularly unexpected finding, the researchers identified that a phospholipid, a type of fat molecule found in cell membranes, plays a crucial role in guiding the receptor's activity, almost acting as a hidden co-pilot. This is the first time such a function has been observed among the more than 700 known receptors of this kind in the human body. Current antidepressants often require weeks to show effects, and scientists are optimistic that this new understanding of 5-HT1A signaling could help clarify these delays and lead to faster-acting alternatives. Dr. Wacker notes, this receptor may help explain why standard antidepressants take so long to work. By grasping how it operates at a molecular level, we can pave the way for designing quicker, more effective treatments, not just for depression, but also for conditions like psychosis and chronic pain. It's a crucial piece of the puzzle. Looking ahead, the research team intends to further investigate the role of the phospholipid cofactor and to validate their lab-based findings through more complex experiments. They are also working on translating these discoveries into tangible compounds that could evolve into future psychiatric medications, building on their previous successes with drug candidates derived from psychedelics. In conclusion, this research not only opens doors for the development of more effective mental health treatments, but also raises intriguing questions about the future of psychiatric medicine. What do you think about the potential for faster-acting antidepressants? Could this new understanding of serotonin receptors change the way we approach mental health treatment? Share your thoughts in the comments. Unlocking mental health breakthroughs. The 5-HT1A receptor discovery. Imagine a world where mental health treatments are not only more effective, but also free of the frustrating side effects that often come with current medications. This vision may soon become a reality, thanks to groundbreaking research into one of the brain's most critical components, the serotonin receptor. Scientists at the Icahn School of Medicine at Mount Sinai have uncovered new insights into how this receptor operates at the molecular level, a discovery that could revolutionize the development of next-generation antidepressants and antipsychotics. The findings, published in the August 1, 2025 issue of Science Advances, focus on the 5-HT1A serotonin receptor, a key player in regulating mood, and a target for both traditional and emerging therapies, including psychedelics. Despite its importance in mental health treatment, this receptor has long been shrouded in mystery. Many of its intricate workings have remained underexplored, until now. But here's where it gets controversial. What if some of the assumptions about how drugs interact with this receptor have been oversimplified all along? This receptor acts like a control panel for brain cells, dictating how they respond to serotonin, a chemical essential for mood, emotion, and cognition, explains Dr. Daniel Wacker, senior author of the study and assistant professor of pharmacological sciences and neuroscience at Mount Sinai. Our research reveals the inner workings of this control panel, how it fine-tunes signals, which pathways it prioritizes, and where its limitations lie. This deeper understanding opens the door to designing therapies that are not only more effective, but also more precise, targeting conditions like depression, anxiety, and schizophrenia without unnecessary side effects. Using advanced laboratory techniques, the team made a fascinating discovery. The 5-HT1A receptor is inherently biased towards certain cellular signaling pathways, regardless of the drug used to activate it. However, the strength of these activations can still be influenced by specific medications. For instance, the antipsychotic asenapine, Sapharis, selectively engages a particular signaling route due to its relatively weak interaction with the receptor. This nuance highlights the complexity of drug-receptor interactions and raises important questions about how we develop medications. To dig even deeper, the researchers combined experiments in lab-grown cells with cutting-edge cryo-electron microscopy, an imaging technology capable of revealing molecular structures at near-atomic resolution. Their work focused on understanding how various drugs interact with the 5-HT1A receptor and how the receptor communicates with G proteins, internal signaling molecules crucial for transmitting messages within cells. And this is the part most people miss. Different signaling pathways controlled by the receptor are linked to distinct aspects of mood, perception, and even pain. By identifying which pathways are activated and how, they can design drugs tailored to treat specific symptoms or conditions while minimizing unwanted effects. Our findings provide a detailed molecular map of how different drugs push buttons on this receptor, activating or silencing specific pathways that influence brain function, says Audrey L. Warren, Ph.D., the study's first author and a former student in Dr. Wacker's lab, who is now a postdoctoral researcher at Columbia University. Understanding exactly how these drugs interact with the receptor allows us to predict which approaches might lead to more effective treatments and which ones are unlikely to succeed. It's a significant step toward creating next-generation therapies that are both highly targeted and safer for patients. One of the most surprising revelations was the role of phospholipids, fat molecules found in cell membranes, in steering the receptor's activity. These molecules act almost like hidden co-pilots, influencing how the receptor functions. This marks the first time such a role has been observed among the hundreds of similar receptors in the human body. Could this discovery unlock new possibilities for faster-acting antidepressants? Current medications often take weeks to show results, leaving patients frustrated and untreated during critical periods. According to Dr. Wacker, understanding the molecular intricacies of the 5-HT1A receptor could help explain these delays and pave the way for quicker alternatives. This receptor might hold the key to understanding why standard antidepressants take so long to work, says Dr. Wacker. By unraveling its mechanisms at the molecular level, we're gaining a clearer path to developing faster, more effective treatments, not just for depression, but also for conditions like psychosis and chronic pain. It's a vital piece of the puzzle. Looking ahead, the research team plans to explore the role of phospholipids further and test their lab-based findings in more complex experimental settings. They're also working on translating these discoveries into real-world compounds that could eventually become psychiatric medications, building on earlier successes with drug candidates derived from psychedelics. The paper, titled Structural Determinants of G-Protein Subtype Selectivity at the Serotonin Receptor 5-HT1A, was authored by Audrey L. Warren, Gregory Zilberg, Anwar Abbasi, Alejandro Abraham, Shifan Yang, and Daniel Wacker. Funding for the study came from NIH grants GM 133504, T32 Training Grant GM 06754, DA 053558, and NIH F31 Fellowship MH 132317. So, what do you think? Should we embrace these new insights as a turning point in mental health treatment? Or are there ethical concerns we need to address when designing drugs based on such precise molecular interactions? Share your thoughts in the comments below. We'd love to hear your perspective.